So tonight we're going to go over Chapter 7. It's on cellular respiration. So on Tuesday we talked about photosynthesis, so how plants make sugar from using the sunlight. So now we're going to talk about how both plants and other organisms use that sugar then to, in essentially using the energy that's stored in that sugar, and in the process, so converting it to the form that they can use in the form of ATP. So mitochondria produce ATP by aerobic respiration. So electron transfer chains in mitochondrial membranes set up hydrogen ion gradients that power ATP formation, similar to what we saw in the light-dependent reactions of photosynthesis. So many disorders related to defective mitochondria are known, including Friedreich's ataxia, in which an enzyme-building protein does not work properly, and therefore enzymes in the mitochondria aren't fully functioning because of this. So this causes loss of coordination, ataxia, weak muscles, and also heart problems. So it's very, very important that the mitochondria are fully functional. They're one of the most important organelles in the body, or in the cell, I mean. So this is an electron micrograph of the inside of the mitochondria. So as you can see, mitochondria have an internal folded membrane, a system that allows them to make ATP, similar to what we saw in the chloroplast. So remember, the chloroplast had interconnected discs or thylakoid membrane. This is a similar idea of what you see in the mitochondrial, the inner space. So you see these invaginations and the inner membrane folded back and forth. So most organisms convert the chemical energy of carbohydrates that they built or ingested into chemical energy of ATP. So anaerobic, without oxygen present, and also aerobic, and oxygen is present. Both of these pathways of carbohydrate breakdown start in the cytoplasm with the process of glycolysis. So glycolysis is the process of converting glucose and other sugars into pyruvate. So this process does not require oxygen. So glycolysis, it doesn't require any oxygen, and if there is no oxygen present, the pathway of cellular respiration ends after glycolysis. So from there you go into fermentation. And so with this, you only yield 2 ATP per molecule of glucose. And we'll get into much more detail on how this works and when you end up using fermentation versus the electron transfer chain. So these 2 terms you need to know well. So first, glycolysis. This is the set of reactions in the cytoplasm in which glucose or another sugar is broken down to 2-pyruvate for a net yield of 2 ATP. So the next one is pyruvate, which is a 3-carbon end product of glycolysis. And fermentation is an anaerobic pathway by which cells harvest energy from carbohydrates to produce ATP. So you have a net of 2 ATP per glucose molecule that's produced using glycolysis. And like I said, we're going to go into much more detail on the process of glycolysis and cellular respiration in these slides. This is kind of just an introduction to what you're looking at. So the first cells on Earth did not use sunlight for energy. So ancient organisms extracted energy and carbon from simple molecules such as methane and hydrogen sulfide. So these are gases that were plentiful in the Earth's early atmosphere. So aerobic respiration, this is something that uses oxygen, this yields much more ATP than fermentation. And this evolved after oxygen was released by early photoautotrophs. And so remember that the oxygen is released during the process of photosynthesis, and that release of oxygen changed the Earth's atmosphere. So the general equation for aerobic respiration is pretty much the opposite of what we looked at for photosynthesis. So glucose and oxygen are converted into carbon dioxide and water. So again, this is the reverse of the general equation for photosynthesis. And this flow chart shows the connection between photosynthesis and aerobic respiration. So the byproducts of aerobic respiration, carbon dioxide and water. So what you see over here. So these byproducts of aerobic respiration are then the inputs for photosynthesis. So these are the reactants of photosynthesis, and the products of photosynthesis are then the reactants of aerobic respiration. So both fermentation and aerobic respiration begin in the cytoplasm with glycolysis. And so this is the slide that gives you a lot more detail on kind of the separation of glycolysis and aerobic respiration. So again, the glycolysis, this begins in the cytoplasm, and then after glycolysis, the pathways of fermentation and aerobic respiration diverge. So aerobic respiration continues inside the mitochondria. And this process ends when oxygen accepts electrons at the end of electron transfer chain. So whereas fermentation ends in the cytoplasm, and in this case, a molecule other than oxygen accepts the electrons. So A in the figure is the first stage, which is glycolysis. And so this is this step here. And so again, glycolysis occurs in the cell cytoplasm. So enzymes in glycolysis convert a glucose molecule to 2-pyruvate, and there's a net yield of 2 ATP during this. Also, you have 2 NAD+, that combine with electrons and hydrogen ions during the reaction to form 2 high-energy NADH molecules. So B, this is the next stage during aerobic respiration. So B, this is the second stage, and this occurs in the mitochondria. Whereas glycolysis occurs in the cytoplasm, the second stage occurs in the mitochondria. And in this stage, the 2-pyruvate are converted to a molecule that enters the Krebs cycle. Carbon dioxide then forms and leaves the cell, and 2 ATP, 8 NADH, and 2 FADH, which is also a high-energy molecule, form during the reaction. And then finally, the third stage is the electron transfer phosphorylation, or electron transfer chain. And this occurs inside the mitochondria. In this stage, 10 NADH and 2 FADH2 that were produced during the previous reactions give up electrons and hydrogen ions through the electron transfer chains. The electrons flow through the chains and set up hydrogen ion gradients that we saw before in photosynthesis, and these hydrogen ion gradients drive ATP formation. And then finally, oxygen accepts the electrons at the end of the chain and forms water. So we're going to now go into more detail on each of these steps. So the general process of glycolysis is shown in the slide. So enzymes use 2 ATP to convert 1 molecule of glucose or another 6-carbon sugar to 2 molecules of pyruvate. So in the process, 4 ATP are formed and NADH is also formed. So again, you have an input of 2 ATP to get the glycolysis reaction started, and in the process you make 4 ATP. So therefore you have a net production of 2 ATP. So going into more detail on the steps of glycolysis, so again, I want to remind you the level of detail that I want you to know. It's in the handout that's on B2L. So make sure you understand all of this to that amount of detail. But I want to introduce you to these individual steps of glycolysis and how it works, just so you understand what's going on. So in the first step, an enzyme transfers a phosphate group from ATP to glucose, and this forms glucose 6-phosphate. And so you can see that here. So next, a phosphate group from the second ATP is transferred to glucose 6-phosphate, and the resulting unstable molecule splits into 2 3-carbon molecules of PGAL. And so you see that right here is the PGAL. So at this point, 2 ATP have been invested in the reaction. And so you can see that you inputted ATP at each of these steps. And so at this point, you have an input of energy instead of a production. So next, enzymes attach a phosphate to the 2-PGAL and transfer 2 electrons and 1 hydrogen ion from each PGAL to NAD+. So 2 PGA and 2 NADH are formed. And then the enzymes transfer a phosphate group from each PGA to ADP. So then 2 ATP are formed by substrate level phosphorylation. And at this point, the original investment of 2 ATP has been recovered. So at this point, we have a net 0 ATP produced. And then finally, enzymes transfer a phosphate group from each of the 2 intermediates to ADP, and 2 molecules of pyruvate form at this step. So 2 more ATP have formed by substrate level phosphorylation. And so summing up, glycolysis yields 2 NADH and a net of 2 ATP. Also, you have 2 pyruvate for each glucose molecule. So depending on the cell and the environmental conditions, one of the biggest things is whether or not oxygen is present. The pyruvate at this point either enter the second stage of aerobic respiration, which is in the mitochondria, or it can be used in other ways, such as in fermentation. So looking at the second stage of aerobic respiration, acetyl-CoA formation and Krebs cycle take place in the mitochondria. So a mitochondria's inner membrane divides its interior into 2 fluid-filled spaces. So again, this is very similar to a chloroplast. So you have an inner compartment, which is called the matrix, and then you have that inner membrane space, which is between the inner and outer membrane. So in the second stage, the 2 pyruvate from glycolysis are converted to 2 acetyl-CoA, and then also 2 carbon dioxide. So acetyl-CoA enters the Krebs cycle, and it takes 2 cycles of Krebs reactions to dismantle 2 acetyl-CoA. So at the end of the Krebs cycle, all the carbon atoms in glucose molecule that enter glycolysis have left the cell in the form of carbon dioxide. So there are 8 steps to the second stage of cellular respiration. So first, an enzyme splits each pyruvate molecule into a 2-carbon acetyl group and carbon dioxide. So coenzyme A binds the acetyl group, and this is what forms acetyl-CoA. So then NAD plus combines with released hydrogen ions and electrons, and this forms NADH. Then next the Krebs cycle starts as carbon atoms are transferred from acetyl-CoA to oxaloacetate. And at this point you have citrate being formed, and coenzyme A is regenerated. So then a carbon atom is removed and leaves the cell in the form of carbon dioxide. So you can see we're at this point right here. So also NAD plus combines with hydrogen and electrons and forms NADH. So remember, this is the high energy form of that molecule. So next another carbon atom is removed and it leaves the cell as carbon dioxide. Also another NADH forms. So pyruvate's 3 carbon atoms at this point have exited the cell in the form of carbon dioxide. And 1 ATP forms by substrate level phosphorylation. So substrate level phosphorylation just means anything that's not phosphorylation through electron transfer. So you're making ATP not using the electron transfer chain. So at this point, the coenzyme FAD combines with hydrogen ions and electrons, and it forms the high energy molecule FADH2. Next, NAD plus combines with hydrogen and electrons and forms more NADH. And in the final steps of the Krebs cycle, oxaloacetate is regenerated from another round to combine back with the acetyl-CoA. So this is a cyclic reaction. So at the end of the second stage of the breakdown of 2 pyruvate molecules, you have a yield of 10 reduced coenzymes. So this is the NADH and FADH2. And also you have 2 ATP that have been produced during the Krebs cycle. So the third stage of aerobic respiration is the electron transfer phosphorylation. So this occurs at the inner mitochondrial membrane. So most of the ATP that are produced are formed during this process. So during the electron transfer phosphorylation, this is when most ATP is made. So in electron transfer phosphorylation, coenzymes reduced in the first 2 stages. So this is the NADH and the FADH2. These deliver their electrons and hydrogen ions to the electron transfer chains in the inner mitochondrial membrane. And electrons moving through the chains release energy, and the energy is used to move hydrogen ions from the matrix to the inner membrane space. So this is using these the energy to essentially push hydrogen from inside to this middle area right here. So the hydrogen ions that accumulate in the inner membrane space form a gradient across that inner membrane. So hydrogen ions follow the gradient back to the matrix through transport proteins, which are ATP synthases that drive ATP synthesis. So again, you have a lot of hydrogen ions in this inner membrane space, and they travel back down their gradient through this ATP synthase. And the ATP synthase uses that energy to combine ADP and phosphate and form the high energy molecule ATP. Also, oxygen combines with the electrons and hydrogen at the end of the transfer chains, and this is what forms water. And so you can see that here. So oxygen is the acceptor of the electrons, and again, this is the consumption of oxygen in cellular respiration. So you're only going to have this electron transfer phosphorylation in the presence of oxygen. You can't have it in an anaerobic environment. The overall aerobic respiration typically yields 36 ATP for each glucose molecule. So you're going to have about 32 ATP that form in the third stage versus 4 in the other 2 stages. But the total yield does vary by cell type. So for example, the typical yield of aerobic respiration in brain and skeletal muscle, these form 38 ATP instead of 36. So there is some variation, but in general, you're going to have 2 during glycolysis, 2 during the Krebs cycle, and then 32 during electron transfer phosphorylation. So what happens if there's no oxygen present? So, situations where you've exercised too much and you're not taking in enough oxygen to support aerobic respiration. So in these situations where there's either not oxygen present or there's not enough oxygen present, you have anaerobic fermentation. And so this begins with glycolysis and it finishes in the cytoplasm with either one of 2 types of fermentation. And so there's alcoholic fermentation and lactate fermentation. So the process of fermentation has a molecule other than oxygen accepting the electrons at the end of the reactions. So alcoholic fermentation is an anaerobic carbohydrate breakdown pathway that produces ATP and ethyl alcohol. So this is ethanol. So this process is used in wines and breads, whereas lactate fermentation is the anaerobic carbohydrate breakdown pathway that produces ATP and lactate. So this is the process used in cheeses and pickles. So this is also the process used in our muscles when they don't have enough oxygen when you're exercising. So the final steps of fermentation regenerate NAD+. So this is required for glycolysis to continue. But in the situation where you're regenerating NAD+, you don't have any more ATP produced. So without the regeneration of NAD+, glycolysis can't continue. So therefore you would stop glycolysis if you don't regenerate this molecule. And so you would stop making any ATP. So the cell's better off using the fermentation process if there's too little or no oxygen present, even though it loses out on the additional 34 ATP that are produced through aerobic respiration in the mitochondria. The breakdown of one glucose molecule in either of the fermentation pathways, alcoholic or lactate fermentation, yields a net of only the 2 ATP that's formed during the glycolysis reaction. But at least this is enough energy to sustain single-celled anaerobic species like bacteria or yeast, or it's enough to at least maintain the processes in the cell for a little while. The skeletal muscle has 2 types of fibers, red and white, and this is in animal cells. So ATP is produced primarily by aerobic respiration in the red muscle fibers. And these muscle fibers sustain activities that require endurance. So the red appearance, which causes the dark meat that you eat on Thanksgiving, is from myoglobin. So this is a protein that's similar to hemoglobin in the red blood cells, but it concentrates oxygen in your red muscle fibers for aerobic respiration. So lactate fermentation in the white fibers supports activities that occur in short, intense bursts. So this is the white meat, such as in the turkey breast meat. So people are born with different proportions of white and red muscle fibers, and so this makes them more suitable to sprinting or long-distance running. So in the end, a lot of, especially your professional athletes, they found that they're born with different proportions of the white or red muscle fibers that make them better at either the long distance running or the sprinting. And so usually you are not good at both. So your body uses the energy stored in chemical bonds of most of the foods that you eat. So simple sugars from carbohydrate breakdown, glycerol and fatty acids from fat breakdown, and the carbon backbones of amino acids from protein breakdown can all enter aerobic respiration at various reaction steps. So when energy demand is low, ATP concentration rises and liver and muscle store glycogen. So when glucose levels decline, the pancreas secretes hormone glucagon, and this causes the liver cells to convert glycogen back into the form of glucose. So this is the first energy reserve that your body taps. So glycogen makes up about 1% of an average adult's total energy reserves. So your body gets energy from fats you ingest or that you have stored. So when blood glucose gets too high, the excess dietary carbohydrates are converted to fatty acids. And the body stores most fats as triglycerides. And so these have 3 fatty acid tails. And so when blood glucose falls, triglycerides are used to produce energy. So the glycerol is converted to an intermediate of glycolysis, and the fatty acids are converted to the acetyl-CoA that enter into the Krebs cycle. And when you eat more protein than your body needs, the amino acids are broken down, and the amino group is removed and becomes ammonia. And the carbon backbone is split, and acetyl-CoA pyruvate, or an intermediate of the Krebs cycle, is formed. So any of these molecules can be used for energy by entering the aerobic respiration pathway. So that's the end of this chapter. We're actually already done with what we're going to cover tonight. I'm going to stay on for just a little bit, and if there's any questions, please let me know. And like I said, make sure to focus on what's on those handouts as far as how to study. That's right. Next week is spring break, so I'll see you in a couple weeks.